Let's welcome our first speaker, Dr. Vittorio Sebastiano. Dr. Vittorio is a leading expert in aging research. He's an associate professor at Stanford University and co-founder, chair of the Scientific Advisory Board, and head of research of Turn Biotechnology. His groundbreaking work on epigenetic reprogramming of aging has earned him prestigious awards and global recognition, including AFAR Junior Investor Award and the Glenn Foundation Breakthrough in Gerontology Award. Please welcome him.
Well, good afternoon. I'm Vittorio Sebastiano. I want to thank Christine for the kind invitation, and of course Eric and the whole Bach Institute for having me. It's always great to be here and to be talking in this iconic institution. So yeah, today I will be talking about kind of what really drives my research and what really drives then Turn Biotechnology's research, which is this kind of concept that is revolving around the idea that we can really leverage reproductive biology for the purpose of finding solutions for aging-associated diseases. And so for the sake of today's talk, as already mentioned, I'm co-founder, head of research, and SAB chair of Turn Bio. And today I will kind of talk about a little bit of my academic work and maybe give you a glimpse of what Turn is trying to do. So I go straight to the point. Ron John was actually alluding to it this morning. He was asking, what is aging? To me, really the most important question is what is really the fundamental linchpin of the aging process at the cellular, tissue, organismal level. And the answer is that I strongly believe that aging fundamentally is an epigenetic problem. So it's something that happens in the epigenome, and because of this deterioration of the epigenetic information, we do see the manifestation of aging, again, at the cellular, tissue, organismal level. It's depicted here in a very overly simplified way. So you know the epigenome is responsible for making sure that actually cells that are genetically identical in your body behave so differently in different tissues. Now, with time, it happens that this program becomes dysfunctional because of a number of reasons. Of course, there is a developmental aspect to it, but then on top of it, there is the so-called exposome, meaning that our cells are exposed to insults that trigger a cascade of events that eventually end up modifying the epigenome that you see depicted here in this, again, oversimplified version. So genes that are supposed to be off or on when the cells are youthful become aberrantly activated or silenced because of the epigenetic information loss. Now, why do I believe it's the fundamental linchpin? Because the epigenome really is the way by which cells behave. So the epigenetic program is the program that dictates cellular behavior. Of course, as a function of that, changes in the epigenome result in changes in the whole spectrum of the hallmarks of aging, right? Okay, so why do I care? Well, I care because, fortunately, the epigenetic program can be reset, can be reprogrammed, because it's an analog program. It's not a digital program like the genetic one, which can also be reset, with CRISPR-Cas9, for example. How? Well, how? The reason of the title of my talk, I think that we just simply need to piggyback and learn what nature has figured out. Nature has figured out a way to not age. The problem is that nature doesn't really care at the individual level if we are immortal. What nature cares is that life is immortal. It's the preservation of life that is important. Now, if you think about this, aging is not vertically transmitted across generations. Again, Ron John said that this morning. Every time we have babies, the babies are not born with the chronological or the biological age of the parents. They're born with an age 0. If you think about that over and over and over across generations, at the cellular level, immortality already exists, because we are the end product of life that is 3.6 billion years old. It's not immortality, but it's very close to immortality, I would say. So, at the cellular level, immortality exists. Ways to not age exist. the question is, how can we piggyback that phenomenal process and translate it to the body? That's the question, okay? And I think really that the answer is in studying women's reproductive physiology and reproductive biology, because the secrets of these are in the embryonic reprogramming, are in the egg, the oocytes, and so I think that understanding women's biology, women's physiology, and women's reproductive biology gives us a tremendous number of opportunities actually to develop anti-aging interventions. All right, now, my lab developed now 11 years ago a technology that is called ERA, that stands for Epigenetic Reprogramming of Aging. It also goes by the name of partial reprogramming or transient reprogramming, but I prefer to call it Epigenetic Reprogramming of Aging because I think it's more accurate. And it was really inspired by, again, nature, reproduction, but practically speaking was inspired by two giants that you see here, Shinya Yamanaka and John Gurdon. So what they did was, now we can say simple, but it was not back then. So what they showed is that you can take any aged somatic cell, and by somatic cell nuclear transfer in the case of John Gurdon, or expressing a cocktail of factors in the case of Shinya Yamanaka, you can actually take those cells and basically bring them all the way back to an embryonic-like state. And this was, of course, these discoveries were given the Nobel Prize in Physiology or Medicine in 2012. Now, what was not realized before, though, is that by doing this process, it's true that you're turning, for example, a blood cell into an embryonic-like cell, but you're also turning an aged cell into a very youthful cell, almost age 0, okay? So what we hypothesized in my lab was, well, what if we could actually decouple those two processes, and what if we can retain the cell identity? So we don't want the cell to become embryonic, because it has some issues when it comes to translational medicine, potential issues when it comes to translational medicine. But what if we can just change the age of that cell? So can we tweak only the epigenetic information that is responsible for age, but not tweak the information that is responsible for cellular identity? So that was the question. Again, we started this at the end of 2014. We published the paper in 2020. And the 11 years after, I can tell you that, yeah, it works. So it's possible, so you can decouple those two processes. So we published the paper. You can just look it up if you wish. So in a nutshell, what we did was to show that you can take any old dysfunctional cell, and by providing the cells with a cocktail of 6 mRNAs that encode for reprogramming factors, and it's important that you control that expression in dosage and in duration. You end up with the same cell type, but just a more youthful version of it. And by doing that, again, since the epigenome, which is the one that we are reprogramming, is responsible for the physiology of the cells, we have shown that actually this process impacts the vast majority of the hallmarks of aging. So the cells are molecularly, epigenetically, and most importantly, functionally youthful, but they're still the same cell types, okay? We have shown these now on more than 12 different human cell types. So we only work with human cells for the most part because I want to develop something that really makes sense. And if it gets hundreds of millions of dollars of investment, then it has a chance to work, very honestly. So that's what we're doing. And I'm actually particularly proud of the fact that actually this field is really booming. So there was a seminal paper by Juan Carlos Belmonte in 2016. He did it in progeroid mice, so a transgenic model of accelerated aging. We were the first ones in humans, but the field is just exploding, and it's really getting a lot of attention from investors, so because I truly believe that this could really be a groundbreaking technology that could be used in the future and really redesign the way we think about rejuvenation. Okay. Just to give you a quick example, so in this case, this is published. So what we did here, we took, in this case, mouse stem cells from the muscle. We isolated them. This was done in collaboration with Marco Quarta, who I think is going to be here soon. And what we did was to take the aged stem cells from the muscle, rejuvenate them or not with this cocktail of factors, just for 2 days, so it's a very short treatment. And then the cells were retransplanted into an age-matched host. And the cells were also, the muscle, the receiving muscle was also challenged, meaning that it was injected with a cardiotoxin to basically physically destroy the muscle so that the stem cells could repopulate and regenerate the muscle at the same time. And what it's really, I think I'm showing you this because it's one of the most stunning results that we got. So this is, you see, and then we took the muscle a month after. You see here that this is the average force that a young, untreated, untouched muscle has. This is the average force that an aged, untreated mouse has. Now, if you take that aged mouse, you transplant age-matched stem cells that have not been treated, you restore the physiological force of that muscle. But if you do the same experiment with age-matched stem cells that have been treated with air, you restore the physiological force of a young muscle, which is pretty significant. It's about 40% of force induction. So where do we want to go from here? Well, and now I switch to Turn's data. Well, obviously the desire now is to really bring this to people's fruition, to the clinic. And how do we do it? Well, one way of thinking about this would be, well, maybe we can think about a way to do it systemically. Systemically, not in the sense of treating every cell in the body, because I think it would be a very bad idea. Some people are doing that, and I strongly disagree with that approach. But doing it in cells that maybe systemically have an impact. For example, immune cells. So here you see a glimpse of data. It's a very busy slide, I'm sorry. So we took aged T cells. So we have shown, and this was done with electroporation, so we have shown that we can deliver and express all the 6 factors that we're dealing with. As a result of ERA, so this transient expression of these factors, we did see a few things happening. Well, first of all, the expression level of these 2 markers was increased. Now, these 2 markers are associated with functionality of stem cells. The higher the expression, the more likely they can kill tumor cells in the body. Okay? So the expression of these was also quantified here. So this basically what this graph shows you is that with time the T cells tend to become effector cells, so very differentiated cells because they also age in this process. When they're youthful and functional, they have a lower percentage of those cells, but they have more stem cell-like cells. So what we have shown here is that with the ERA treatment, we can restore or enhance the population of stem cells or stem cell-like cells in the population of T cells, which is, again, from a diagnostic standpoint, has a potential to be more effective. The other important thing that we have shown is that the cells have a higher proliferative capacity when put together with cancer cells, with the target cells. So you see here that in response to ERA, and ERA in this time was done for 3 days or 6 days, the proliferation of the T cells is enhanced when they are engaged with the tumor cells. What does this mean? Well, it means, just another thing. So this from a molecular standpoint, this shows that ERA can really impact the T cells population and rejuvenate them, giving them higher proliferation and potentially higher functional activity. What happens to their identity? You remember that I said we don't want to make embryonic cells. We want to keep the cells as they are. Well, we have shown this at the molecular level. Every single marker of T cells is not changing, meaning that molecularly the cells are still the same cell types. Okay? And this is fantastic for the publication, but what I care at the end is well, what about functionally? Because that's what we care about. And functionally, they're also not only preserved, but enhanced in their functionality. So you see here on the left, the green cells are the cancer cells, so the cells that we want to kill. On the left, you see that the cells that are untreated at some point exhaust, they become fatigued and aged, and they can no longer kill the cancer cells. On the right, you see that actually with ERA, the cells overproliferate and they get rid of the cancer almost in no time. And keep in mind that this is the 3rd consecutive engagement, meaning that the cells are constantly challenged with fresh, new cancer cells in order for them to really be super challenged. So again, you see on the left, they exhaust and they die at some point. On the right, they get rid of the tumor cells. So now the point is to try to make in vivo data building on this. So another thing that we are trying to do, and then I'll close the turns part here. Oh my God, I'm really late. Is that we're now trying, well, we have done this work with the T cells. What we want to try to do is now to work on the blood stem cells. And so just today we announced a partnership with the Children's Hospital of Philadelphia to actually start this preclinical work. Okay, I'm doing really late. So what I wanted to tell you is that I go back to the publication, when we looked at the epigenetic clocks in fibroblast and endothelial cells, we have shown that actually ERA can truly rejuvenate, also using epigenetic clocks as a way to measure aging, was shown to have an impact on the epigenetic clocks. But what was surprising is that when we start digging into the data, we discovered that actually that rejuvenation was actually explained by one single CpG in the whole genome, which is about 7 billion base pairs. So it was explained by one single base in the genome. And that was just stunning to us, because we were expecting actually that this reprogramming was actually much, much, much broader and much more impactful on the chromatin. So I really couldn't wrap my mind around the fact that how is it possible that we have such a drastic rejuvenation, and with clocks we see only one CpG really changing significantly. So this prompted us to develop, I'm going to skip this, but to develop basically a new way to look at this problem. And this work was just recently published and was spearheaded by Madi and Andrea. Andrea is in the audience somewhere. He's a very tall guy, so very easy to identify. Basically what we started was to say, well, maybe we should start looking at this problem in a completely different way. Instead of looking at just a handful of CpGs, we should start looking at the whole genome and really try to understand the biology of this process. To understand aging, but most importantly, to understand the effect of rejuvenation in different ways, using different treatments and so on. So what does the rejuvenation do to the epigenome? And do all the rejuvenation technologies, work? So I have to really go quickly here, but basically to make a long story short, we have developed a new index that is called PRC2 index. And what we have shown is very simple. We have shown that there are some specific sites in the genome that have a high binding affinity for PRC2 complex, which is a repressive complex, that with time gain methylation. Okay? So you see them here. So these regions that have a very high binding affinity for PRC2, over time, they gain methylation. Why is this important? Well, it's important because, and I want to stop here, it's important because we have discovered that those same regions respond differently to different rejuvenation interventions. So, for example, look at this graph here. We have shown that the rapamycin treatment, for example, does not seem to affect those regions. It actually seems to even enhance the methylation of those regions. Caloric restriction, on the other hand, seems to impact those regions, and so it seems to kind of erase the methylation of those regions and bring it back to a more youthful state. And the same is true for ERA. So this is not our data, but it's data from Juan Carlos Belmonte that we just reanalyzed. You see that those regions become demethylated with the partial reprogramming, similarly to what happens with caloric restriction, but not, like what's happening with rapamycin. So there is a phenomenal biology there to discover, and I think we are really now starting to understand not the correlate of aging, as Ron John was saying, but the causative changes of the epigenome on aging. Okay, and I think I want to stop there. Sorry, I had to rush through. I just want to thank the people in my lab, the funding, my institute, you obviously for your attention, and just 1 minute to say that I am together with Eric and many other people, Jamie and so on. We have been putting together the Biomarkers of Aging Consortium, which is a very, very exciting consortium that is trying to gather the people that are working on biomarkers to really come to something that is really useful and to a consensus, actually, because there's so many biomarkers and we're having a hard time understanding what to do with them. So the consortium is actually is a really an entity, non-profit, that is trying to gather people to work together on this exciting project. We are having the third annual conference in Boston in October, so please sign up because it's really a good meeting. And there is also an ongoing study that will be launched officially. It's a longitudinal study that we just closed the pilot, and now we're going to launch. So this is going to be a longitudinal study on a variety of different biomarkers on people longitudinally, very important. So if you want to participate, if you want to sign up, just go to the website and check it out. Thank you, and sorry for being so quick.
Hi. Very nice talk. I was more interested into the T-cell rejuvenation that you mentioned, actually. So I hope this is in vitro data that you are showing, or have you done in vivo studies using the ERA treatment in any mouse experiment or something?
So yes, you're right. The data that we have shown is all in vitro for now, because we want to really thoroughly characterize the process and make sure that the cells haven't changed molecularly and functionally their identity. We have shown functionality enhancement in vitro. The next step will be to do in vivo studies, but we haven't done that yet.
Yeah, I'm very surprised to see if ERA treatment can bring back exhausted T cell into a naive T cell population. What happens to the other T cell subtypes? Have you done any kind of a deep immunophenotyping?
Not yet. All we know for now is that we have looked at a variety of different markers. So I've shown only a small number of them. Across all of the markers that we have seen, so again, we don't see cell identity change. We do see change in the population, relative population. So we see more stem cell memory and stem cell effector cells, and less effector and effector memory.
Yeah.
And this has already been shown in the literature to have potentially a higher therapeutic potential in the patients. Now, having said that, this is just speculation for now. We need to show it. We need really to do the in vivo data.
Yeah, my last question is, if ERA can bring back the naive T cell population, can this be used as an in vitro immune model? Can the ERA treatment can be used as an in vitro immune model, what the entire scientific community is trying to build to study or screen any kind of rejuvenation treatments?
Yeah. I mean, I am biased here, so take it with a grain of salt. ERA, as we have shown it, and many other people are working on it in different flavor of it, ERA can work theoretically on any cell type. The question is to try to figure out the best way to express the cells, and I'm a strong believer that this should be done with mRNAs, but again, I'm biased, so take it with a grain of salt. And try to figure out what is the duration of the reprogramming, because that's the key of the technology.
So beautiful data. Out of curiosity, on that note of duration, how do you modulate duration of your therapy? I know you're doing electroporation, but is it the duration of the electroporation itself, or is it?
No. So I have shown the data with electroporation, but no. In the company, we're also developing LNPs to deliver in vivo the RNAs. The duration is dictated by the number of transfections and by the half-life of the mRNA that can be in silico designed and in silico optimized to last only a certain number of hours and not more.